Okay guys, in this particular lesson we are going to be talking about changes that occur in genetic information and what those changes actually mean for the human body. So the first thing that we're going to talk about is the fact that the nice thing about your genetic information is that your DNA has the ability to proofread itself. Basically it has the ability to go back, double check, and make sure that the base sequence is correct. And if it's not, it has the ability to actually go back and fix those mistakes. It's called DNA repair. DNA has the ability to repair itself if necessary. But unfortunately, there are some mistakes that slip through the cracks, and those slips that slip through the cracks, we call them mutations. Mutation is basically just a change in genetic information, and we typically associate that change with being a negative, although that is not necessarily always the case. Sometimes mutations can be positive. But that mutation is basically an alteration in the DNA base sequence. So there can be a lot of different causes for why mutations may occur, why the DNA base sequence may be incorrect. We know that during replication, the DNA base sequence has to be copied. It needs to be read, and complementary base pairs need to be lined up appropriately, and this can be a tedious task, and every so often an incorrect base can be paired, extra bases can be added, sections could be missed, sections could be misplaced, and obviously the result would be that the base sequence would then be incorrect if these mistakes are occurring during DNA replication. So the resulting DNA molecule would have a mistake somewhere. There are also things called mutagens, which are things in the environment, things such as chemicals, radiation, sun exposure, etc., all these environmental factors that can alter the genetic information, the DNA base sequence as well. This is just a nice little picture that shows you types of mutations and showing you that there is basically a sequence that exists. So in the very top it says that the normal gene, and I'm just going to draw a line through here to kind of see how we're dividing. The normal gene is as the man saw the dog hit the can end. But a point mutation would be if one single mistake existed. For example, the G became a T instead. That little point mutation, if that little point mutation occurs, well then that means that this triplet in DNA is going to be coding for probably an incorrect amino acid maybe, and if it's an incorrect amino acid, maybe that's going to have an effect. So instead it's as the man saw the dot hit the can end. Versus a deletion mutation. In the case of the deletion mutation, we've deleted the entire word dog, or dot, however you want to look at it, the whole word dog, so it's as the man saw the hit the can end. We're missing that whole portion right there. An insertion, as the man saw the fat dog hit the can end. All right, so we have something was inserted, extra genetic information. And then a frame shift, as the man saw the, oh, we have a deletion. Do you see this? There's a deletion right here, and as a result of the deletion, there's a whole shift that occurs. Right? Our triplets, because only the D is deleted, our triplets, everything shifts over one. So as the man saw the og, it, heck, ain, nd. And then you're kind of left with a missing space. So this is just an interesting way to look at how these mutations can alter the information that you find. But, like we said, there are protection mechanisms in place. There are ways that the genetic information is protected to ensure that it's properly carried out. And those are with the repair enzymes, where DNA errors are detected, they're removed, and replaced with the correct nucleotide. This is that DNA repair that I just mentioned. All right, where enzymes will actually go in and they'll chop out the little mistake and they'll fix the mistake to hopefully protect the genetic information, the genetic code, so that the proper proteins can be created during protein synthesis. In addition, there are actually 61 codons that exist for only 20 amino acids. Obviously, that means that amino acids, many of our amino acids, have more than one codon coding for it. Like we've got doubles and triples of codons that code for the same amino acid. If a mistake is in the third base in a triplet, oftentimes it doesn't change the amino acid sequence. And a lot of times, if it's in the second base, it's coding for an amino acid that's substantially similar to what it should have been. What I mean by that is if we look here, all right, at this section of our table for proline, the proline amino acid, it doesn't matter whether the genetic code says CCU for the codon, CCC for the codon, CCA, or CCG. So long as CC are the first two, they're all going to code for proline. Similar for threonine, leucine. Even further than that, leucine, look at the combination we have there. For leucine, there's a pretty substantial number of codons that can code for the same thing. This like doubling up and tripling up kind of protects the idea that if a mistake happens, oftentimes the mistake is not detrimental, right? So being able to, anything that's GU, something will be valine, GC, something will be alanine, and etc. That's kind of one of our protective mechanisms. But unfortunately, mutations can occur. So what you have here is, as we can see depicted by the red, is that we have an error, right? We have a single point mutation is what it's called in a base sequence. So at the top is what you would want the actual base sequence to be, and at the bottom is the mutation. And as you can see, these are codons 4, 5, 6, and 7, so you still have 1, 2, and 3. And what actually happens is that when this one single adenine molecule is changed into a thymine molecule, what actually occurs is sickle cell disease, which I'm sure you guys are familiar with. Sickle cell disease occurs when the 6th amino acid is altered. So obviously, if what we have here is the 6th codon, that means it's going to be the 6th anticodon, which means it'll be the 6th amino acid in the chain. And when that 6th amino acid is different, based on this one single base error, hemoglobin, the protein that's supposed to be made, is abnormal. And if the hemoglobin is abnormal, then the hemoglobin ends up bending the red blood cells into that sickle cell shape. And that sickle cell shape then becomes more likely to clog marrow, blood vessels, and become a problem throughout the body. So even though I'm pointing out that, there are many mechanisms in place to protect against having differences, there are some occasions where one single little difference can be a relatively big one. These are also some commonly encountered mutagens. I don't know about you guys, but that one kind of worries me. You guys know how much I love my coffee. And it is a mutagen. All these different things. Hair dye. All right, components in hair dye are mutagens. We know cigarette smoke, smoked meats. So there's a variety of, and these are just commonly encountered ones, guys. These are things that are relatively common for our population. Dyeing hair is really common. Drinking beverages with caffeine, very common. Using pesticides, plants having herbicides on them, things like that, where you're ending up ingesting those later if you're not carefully washing your produce. We've talked, even if you do wash your produce, you're still probably ingesting some of these chemicals. Flame retardant chemicals in children's sleepwear that you probably would never even think of. These are just commonly encountered ones, guys.